Good morning, dear colleagues. My name is Sarah Jufaili from Ministry of Agriculture, Fisheries, Wealth and Water Resources. On today's presentation, I will be talking about some of the findings of our ongoing research projects investigating the occurrence and abundance of zoonotic anisakid nematodes in the abdominal cavity of selected edible fish species from Oman. Since the establishment of the Laboratory of Aquatic Parasites in 2009, our team was involved in several research projects aimed to survey the occurrence of various fish parasites in Omani water. Seafood safety has been recently become a great concern for the consumers. That's why our main focus is on aquatic zoonotic parasites, which are potentially transmittable and able to survive in the human digestive tract after consumption of raw fish infected by larval helminthes. So next slide. So what are anisakid nematodes? Anisakid nematodes are parasitic worms with complex life cycle. Um, they have a worldwide distribution, and they are known to infect different aquatic hosts, like marine mammals and seabirds and fish hosts. They are utilize different tropics food levels in the marine system. So the larval stages of these worms are some of the known to cause significant problems for the commercial fisheries industry and could be a potential human health hazard or as food allergens. So despite the importance of fisheries sectors in Oman, and its high marine biodiversity, there is a lack of information regarding zoonotic parasites in Omani water. That's why this is the importance of our study. So Anisakid nematodes is a huge family of worms, but we are concerned about only 3 genera of these worms. As you can see, the Anisakis in the picture in the right top, and then in the picture in the bottom in the left picture. So these are different types of worms that can be found in hosts and seabirds. So why they are important is because they are known to cause clinical disease called anisakiasis. In statistics, it has been reported that 20,000 cases of human anisakiasis infections are reported every year, especially from countries, coastal countries around the world. So anisakiasis or anisakidosis is a disease that is caused by these worms. This disease occurs usually by ingestion of raw fish or undercooked fish that are infected with these worms. So humans are actually accidental hosts. They are not the main host for these worms, but they could be accidental. So in the right, you can see the life cycle of the parasite, and the red arrows are the normal life cycle of these worms. And you can see that the dolphins are the main definitive host, or other mammals. But when the human consumes fish that are infected with these worms, they become also as hosts. So how can they consume raw or undercooked fish? We know that some countries, they have popular traditional foods like sushi, sashimi, and other pickled or raw fish. From these products, if they are not checked well, they could transfer the worms into the human. So the symptoms of the infection could include epigastric pain, nausea, and vomiting. And in some severe cases, it could be like allergy reaction. So for our work, for our research purpose, we collected 1,680 fish from different locations and from different landing sites around the Sultanate of Oman. Once the fish is sent to our labs, we will start by dissecting the fish, and we will conduct visual and microscopic infections. We make sure that we collect fish from as much landing sites as possible to cover all the regional coastal line of Oman. When we finish our inspection, when we detect the parasites, we will take them from the fish, and then we will prepare them by fixation to conduct different methods like the light microscopy, scanning electron microscopy, and PCR and sequencing. So these are the type of fish that we have collected. So far, we try to include demersal and pelagic fish that are commercially important in the Sultanate of Oman. So once we detect the fish, as I said in the previous slide, we will prepare them for morphological investigation, because the worms that we find usually in the fish, they are from stage 3 larvae. So they have specific characteristics to be able to classify them into their respective genera. So for accurate identification, it is really important to know these features and to differentiate them. So that's why we are using different methods like the light microscopy and scanning electron microscopy to study them. And in some cases, we also use confocal microscopy. However, identification to species level is not enough only by morphology. It is necessary to follow up with genetic identification to confirm the species of the worms that we have detected. So for molecular investigation, we extract the DNA using commercial extraction kits, and then we will perform the PCR reaction using the ITS region, which is the most common region used for anisakid nematodes. Once the reaction is finished, we have to confirm it using the gel electrophoresis, as you can see in the right image. And then when we get the positive reactions, we will send them for sequencing to confirm them further. So these are our results so far. So the project is still ongoing and we are still getting new results. So this is not the final result, but so far we have a total of 9 species of anisakid nematodes from Omani fish. And the most common one is Anisakis typica, the second one on from the left. And compared to literature from other countries and from other fish hosts, the infection intensities and occurrence in Omani fish is the lowest so far. Also, we have to know, I will check the second slide here. This is the distribution according to the hosts. So as I said, we examined 19 hosts, and you can see here that some hosts, they didn't have any worms in them, and some hosts have only 2. Other hosts have more than 3. So it depends on the type of hosts. This is a summary of our findings. It is in a summary, we have to know that the most common is Anisakis typica and Anisakis faceritis, and the highest diversity is found in oblique-banded grouper and ribbonfish. No anisakids were found in needlefish and black-tipped trevally and lizardfish. Also, it is important to note that most of the parasites were only found in the abdominal cavity of the fish. This is very important because we know that anisakid nematodes that are found in the flesh are the dangerous ones, and the ones which are found in the abdominal cavity, they are not of great concern for human health. However, this will suggest us to inform and educate the fishermen and the consumer to take care to avoid the migration of parasites from the abdominal cavity to the flesh. This we will talk about it in the end of this presentation. For the genetic confirmation, this is just a picture to show that most of our PCR products were successful and successfully amplified. And this is the results we obtained from the International Gene Bank to confirm the species that we have found so far from Omani fish. So for the regulations and policy, the purpose of this study is to have an idea about the occurrence of these worms in Omani fish, because we are lacking of this information. For the purpose of exportation and the local consumption, we have to know what we have in our fish. Because if we are just relying so far on FDA regulations and EC regulations, which are not reflecting the situation that we have in Oman, because they are dealing with different types of fish due to their geographical region. So far we are using the same regulations, but in the future, as we proceed with our research programs and we have more information from our fish, we will be able to make our own policy and update our regulations. So for the regulation, it is suggested that to freeze the fish that have been found to be infected with these worms for at least 15 hours, or in some cases no less than 24 hours. And if we find that fish have more than 20 nematodes per kilo, this is in case of salmon. That's why we have to apply our own regulations in the future. We will consider this fish as not suitable for human consumption. So based on our findings so far, we suggest for the consumers to make sure that when they buy the fish that are supposed to be infected or are suspected to be infected with these worms, to try and transport them in chilled status. So they have to put ice on top of the fish to avoid to reduce the activity of the parasites, because the parasites, they can be active once the fish is dead. The parasite in the abdominal cavity tries to escape to the outside to find a new host, because the current host is dead. That's why they try to escape. So when they try to escape, they migrate from the abdominal cavity to the flesh. So to avoid this migration, we could either use the ice to suppress the activity of the parasite, or to remove the viscera in capture. So we have to also suggest to the fishermen, when they capture the fish which are known to be infected with these parasites, to remove the viscera immediately, or to also suggest to the consumers to remove the viscera before storage. And for cooking, in Oman, we don't have a problem with cooking fish because we usually eat fried or baked fish. But in some cases, if the restaurants, they want to use for sushi and other pickled or smoked products, it is suggested that the fish should be cooked in temperature at 60 degrees. Also, the fish that are suspected to have these parasites should be frozen at -20 for 24 hours. In case of salting and pickling, it is suggested that heavy salting is suggested for this kind of product, at least for 10 days, to make sure that all the live worms are dead. So that's it for my, this is concludes